Hello students, in this video I would like to describe about the structure of seeds and growth of plants. Dear students, if you observe a seed of bean, then you will see it's shaped like an egg, right? So a seed may be two parts or a single part depending on the types of the seed. So here we have a seed of bean which is in two leaves. These are called cotyledons. Okay, these are called cotyledons. And outer covering of the seed is called seed coat. There is one eyes. There is one eye present on the leaf which is called micropyle. What is called? It is called micropyle, through which teeny baby plant grows. This is the teeny baby plant present here to show from the micropyle a teeny baby plant grows. So take some gram or bean seeds, soak them for 10 to 12 hours in water. Now pick one seed out of these and observe its structure carefully. You will notice that it has a hole that allow water to enter its inside. This small hole is known as the micropyle. The seed is covered by an outer layer called seed coat. So this is the outer covering which is called seed coat. Okay. So inside the seed coat there are seed leaves which are called cotyledons. So seed leaves are called cotyledons. Some plants have, like peas, gram and beans, have two cotyledons, whereas wheat, rice and maize they have only one cotyledon. Right? Who have only one cotyledon? Wheat, rice, maize. Their seed cannot be divided into leaves like the bean or gram seeds, right? Because they have single cotyledon. Inside the seed leaves there is a baby plant. Just you see, this is the baby plant inside the seed leaf. Okay, there is a baby plant. The baby plant has a teeny root and a teeny shoot. A baby plant has teeny root and so just you observe carefully that this part is will grow underground, that is called root, and this part of the baby plant will develop upper side of the soil, that is called shoot. Okay, so there are small leaves present here that will develop into shoot. Okay, so the baby plant has a teeny root and a teeny shoot. The the cotyledons store food for the baby plant so that they can grow. Now growth of the plant. The baby plant starts growing into a new plant when it gets appropriate amount of air and water and the temperature around it. So what are the necessary conditions to grow a plant? Necessary conditions are, that is, air, water and temperature. So neither too hot nor too cold. A plant cannot grow in very hot temperature or very cold temperature. To start with, the cotyledons provide food to the growing plant. Later on, the plants start making their own food with the help of their leaves, that is process of photosynthesis. Next step is seed germination. As we have described here the structure of the seed, and we have seen here that a baby plant is present, a teeny baby plant is present in the inside the seed leaves. So how do plants germinate? We know that plants usually grow from a seed. The process by which a seed grows and develops into a seedling, that is young plant, is called seed germination. What is seed germination? The process by which a seed grows and develops into a seedling is called seed germination. Now you see this picture, observe it carefully. This is a bean seed. Now bean seed, here you see that a small opening is coming out, emerging, that is root. So root develops towards the soil, and this root grows under the ground. Then shoot system. Now you see that a shoot system develops into upside of the soil, outside of the soil. That is, these are the cotyledons through which green pigments or leaves are coming out. That is called seedling or young plant. And after that you see that there is young plant is growing on, right? Baby plant is growing on. So in this way you see that roots are developed inside the soil and shoot is developed outside or above the soil towards the air. So this will after developing the leaves, the plant started carrying out the process of photosynthesis, so they make their own food. But meanwhile, as they are teeny baby plant, till then they absorb the food from these cotyledons. Okay, so seeds need favorable condition to grow and develops into seedling. Okay, this do activity. So just you see that in step A we have to do this activity. Step A, soak the some seeds of gram or beans for 10 to 12 hours. Then keep seed without water. Take another set in which seeds are kept in a bowl but without any water in the at the same room temperature. Okay, after 10 to 12 hours you will observe that the bowl in which water seeds are kept in the water, their outer surface means seed coat becomes soft, and if you open it then you will observe that the seed cotyledons can be separated, and then we can see the teeny baby plants as shown here. Okay, now take another bowl and in which you put some ice and put the same amount of seeds in it. So you have the four sets of your bowl. One is the keep the seed on moist cotton and room temperature. Seed in this one, moist cotton. Second is keep the seed without water at room temperature. Third one, keep seed in a dish completely filled with water at room temperature. And fourth is keep the dish having seed on moist cotton in an ice box or a refrigerator. Now you observe that which sample is getting the favorable condition. Here, sample 4 in ice box, it will not get the sufficient temperature. Here, here the seeds are filled completely in water, so they cannot get air. Here, they do not get water because they are kept in a room temperature without water. In this sample A, they get all favorable condition because through moist cotton they absorb water. They get air from outside and appropriate temperature as this bowl is opened. So the plant will grow only through these seeds. Possibility of the growing of plant is only from this sample. To just you see here as a teeny baby plant, that is called seedling. So seedling will take place only through sample A. So after 3 to 4 days or 4 to 5 days, you will observe that seeds present in the glass dish BCD have no germination. Only the seeds of dish A grow into seedling. This is due to the fact that dish A, the seeds present in dish A, they get appropriate water and air and temperature, that is warmth needed for germination. In the other glass dishes, either one or more than one favorable conditions are missing. Thank you.